Have you ever wished you had an owner's manual for life itself? I mean, what if I told you we're getting really, really close to having one? Today, we're going to dive into the incredible toolkit that lets us read, rewrite, and truly understand the most fundamental instruction book there is, our DNA. Let's get into it. Okay, so check this out. You've got two apples, you cut them both open. One starts to turn that gross brown color almost right away, but the other one, it stays perfectly fresh. So, what's going on? It's not magic, I promise you. The answer is written right there in their genetic code, and the fact that we can now change that code is really the heart of our whole story today. So, how in the world do you actually edit the source code of a living thing, like an apple? Well, that brings us to our first big topic, this powerful set of tools we've developed for literally rewriting the code of life.
The key to all this is a process called recombinant DNA technology. Now, I know it sounds super complex, but the idea is actually pretty straightforward. You basically just take a piece of DNA with a trait you want from one organism, and then you insert it into another one. So for our apple, scientists took a gene that switches off the browning enzyme and just popped it in there.
But hang on, how do you get that new gene into the apple's cells? It's not like you can just poke it in there. You need some kind of delivery system, right? In genetics, we call this a vector. Just think of it like a biological UPS truck designed to carry a precious genetic package right to its destination. And you know what the most common delivery truck in this whole business is? The plasmid. It's this tiny little circular piece of DNA that's just naturally found inside bacteria. At some point, scientists had this brilliant idea. They realized they could basically hijack these little rings to carry any gene they wanted into a new cell. So the whole process, when you boil it down, really comes down to four main steps. First, you got to get the gene you're interested in. Second, you get a plasmid from some bacteria. Third, and this is the cool part, you cut both of them open. And then fourth, you just join them together. Okay, so how do you make such a precise cut in a molecule you can't even see? Well, you use something called a restriction enzyme. The best way to think about it is as a pair of molecular scissors. They are totally amazing. They're engineered to cut DNA only at very, very specific sequences. And once you have your gene and your plasmid all cut open, you need to stick them together, right? For that, you use another enzyme called DNA ligase. And if the restriction enzyme is the scissors, then this is your biological glue. It perfectly seals the gene into the plasmid and bam! You've got your new piece of recombined DNA. And this tech goes so far beyond just non-browning apples. I mean, it's absolutely revolutionary. It's used to make huge quantities of human insulin for diabetics, to engineer crops that can fight off pests all by themselves, and even to create the enzymes in your laundry detergent that get rid of tough stains. It's everywhere. But, rewriting the code is only half the story. What about reading it? This very same kind of technology also gives us the incredible ability to create a totally unique identifier from a person's DNA, basically a fingerprint that's written in their cells. I want you to picture a crime scene. A detective finds just a single drop of blood, or maybe one stray hair. In the past, that might not have led them very far. But today, that tiny little sample holds enough information to potentially identify a suspect with just incredible accuracy. Now, here's the part that just blows my mind every time. Genetically speaking, you and I and everyone else on the planet are 99.9% identical. It is that teeny tiny 0.1% that makes each of us unique. And that's exactly where scientists look to create a DNA fingerprint. So to actually see this unique pattern, scientists use a technique called gel electrophoresis. Basically, they put DNA fragments into this special gel and then run an electric current through it. And because the smaller fragments get pulled through the gel faster than the big ones, this whole process neatly sorts all the pieces out by size. And what you're left with is this pattern of bands, kind of like a barcode that's unique to a person. And this is the aha moment. You compare the barcode from the crime scene sample to the barcodes from your suspects. And right here, you can see it plain as day. A perfect match between the crime scene DNA and suspect 3. Case closed. And of course, this isn't just for crime shows. As cool as that is, DNA fingerprinting gets used all the time to confirm family relationships, to identify victims after disasters, and even in conservation to trace illegal ivory back to a specific poached elephant. It is an incredibly versatile tool. So we can rewrite the code, and we can read an individual's unique signature, but what about reading the entire book? That was the goal of one of the most ambitious scientific projects ever undertaken, mapping the complete human blueprint. The Human Genome Project had this one clear, absolutely monumental goal: to sequence letter by letter the entire genetic instruction set for a human being. It was this huge global collaboration of scientists, all racing to read our complete genetic story for the very first time. And listen, this was no overnight success story. The project officially started way back in 1990. It took them almost a decade just to sequence the very first chromosome. But they kept at it, and by 2003, 13 years after they began, the final complete version of the human genome was published for the world to see. So after all that work, what did we find? The numbers are just staggering. The human genome contains about 3.2 billion base pairs. Those are the letters A, T, C, and G that make up our code. That's like an instruction manual that's over 3 billion letters long. But here's one of the biggest surprises of the whole project. Scientists originally thought we'd have maybe 100,000 genes, or even more. The actual number turned out to be around 21,000. It was a real shock. It told us that an organism's complexity isn't just about how many genes you have, but about the incredibly intricate ways those genes are used. And this project confirmed, on a massive scale, what we saw with DNA fingerprinting. The DNA of any two people on this planet is 99.9% identical. I mean, just stop and think about that for a second. All of human diversity, our different looks, our talents, our histories, all of it is packed into that tiny 0.1%. It's such a powerful reminder of how fundamentally connected we all are, written right into our code. So, we've come an incredibly long way. From creating an apple that doesn't brown, to fingering a criminal from a single hair, to reading our entire genetic encyclopedia. We've unlocked an immense amount of power, which of course leads to the really big question: what do we do now? We're standing at this totally unique moment in history. We have the owner's manual. We've got the pens, the molecular scissors, the biological glue. The code of life is officially open for editing. So the question that's left for all of us is, what stories, what cures, what kind of future should we write into it?